Welcome back to Chapter 2. This is the second half of the chapter. We've been talking about virus structure, and I wanted to talk about icosahedral virion structure. So this is arguably the most common viral shape. It's definitely more common than helical that we were just talking about, and that is likely due to the fact that studies show that it's just energetically more favorable when you're talking about, especially talking about viruses that are composed of multiple subunits. And an icosahedron is composed of 20 faces, each made out of equilateral triangle. You can see that in the image here. Here are just a few examples of 3D renderings of icosahedral virion structures of some common viruses. You can see some of them actually look rounder rather than icosahedral, and that's really just due to the fact that there are proteins, typically glycoproteins, extending from the surface that kind of fill out some of the angles and make it look a little more spherical than it actually is. A few terms that are worth noting on an icosahedron. You have the vertices, vertex, which is the points at which the points of 5 triangles come together. So basically the pointy bits here. You have the face, which would be the center of each equilateral triangle, and then the edges, denoted by the red triangles here, actually the edges of the triangles themselves. And so in relation to that, you'll see these terms thrown out as we go through future chapters, especially when they're talking about like the location of specific proteins on the surface of a virion. They might say, oh yeah, that protein has twofold axis of symmetry, or threefold, or fivefold. And what that means is, okay, well, if we say a protein on the surface of this icosahedral virion has twofold axis of symmetry, it means it's located at the center of an edge. Whereas if it has threefold, it would be at the center of a face. If it has fivefold, it would be located at one of those vertices. So you'll see that terminology moving forward. Just want you to be aware of what that meant. So as we talked about, these virions are made up of capsid proteins, and there are no proteins that form equilateral triangles. So it's impossible to have basically like one capsid subunit per face. Research shows that you have to have at least 3 different subunits for a total of 60 to build an icosahedron. And so those subunits could be identical to each other, like 3 identical subunits coming together to make the icosahedron, or they could be 3 different subunits, just depending on the particular virus. Another term that's important here in reference to icosahedrons is the triangulation number, which is basically an indication of the size of an icosahedron. So the triangulation number is the number of structural units per face. So for example, this example, we have a triangulation number of 1 because we have one structural unit. It's actually made of, that one structural unit is made of 3 different proteins, but one structural unit per face, versus triangulation number of 3. We have 1, 2, 3 structural units here for that one equilateral triangle, excuse me. And so again, basically the larger that triangulation number, the larger the icosahedron is going to be. You can see that illustrated here. Triangulation number 1 versus 3 versus 4. They get larger and larger quite quickly. And so that triangulation number is something that can be mathematically calculated. I'm not going to ask you to do that, so I'm not going to spend much time on here, but I did just want you to be aware that, if you know basically how many structural units, how many subunits you've got, you can do a pretty simple calculation and determine that triangulation number for any icosahedral virus. Okay, so we had helical virus structure, we had icosahedral, and then the third category was complex, which, as I said, is basically everything else that's not helical or icosahedral. A few interesting examples here: vaccinia virus, which is related to smallpox. In fact, most of the research that was done trying to understand smallpox was actually done on vaccinia virus. Um, has this interesting like dumbbell-shaped capsid you can see here on this electron micrograph on the interior. Gemini virus, which is a plant virus, has again an unusual sort of two-lobe structure, and we've already mentioned and looked at bacteriophage, right, which have this sort of head and then a long skinny body and sometimes little legs or arms coming off of that. Um, so again, just be aware if it's not helical or icosahedral, we're going to call it complex structure. All right, so to finish this chapter out, I wanted to talk just a little bit about virus classification and taxonomy. And I'm not a taxonomist, so I don't want to spend too much time on this. But you will see this in other chapters being mentioned. You know, anytime they introduce a new virus, they'll mention its taxonomy. So it's definitely worth at least brushing up against this here. Um, so when you're classifying viruses, really can't use standard taxonomic techniques, because those really only apply to living organisms, right? And as we discussed in the first chapter, viruses are not living. They're outside that tree of life, so we have to have unique taxonomy. And there's really two commonly used systems. There's the Baltimore classification system, which is 7 classes based on uh, the composition of the genome. Is it DNA, RNA, single, double-stranded? And then it also looks at some of the details of the specific replication strategy of that virus. So how does it go through its replication cycle? And we're going to see some of these in chapter 4 when we look at some of these different replication strategies. They'll have them broken into this Baltimore classification system. But the other main system is the ICTV, which is short for the International Committee of Taxonomy of Viruses. And this is based on lots of different properties. They also look at the genome, the nucleic acid, look at the biochemistry, capsid, etc. So let's look at this one here for now, since we're going to see the Baltimore system in chapter 4 as we go forward. So this should look somewhat familiar, right? We do see some familiar taxonomic terms being used: order, family, genus, species. But it's worth noting that for viruses, order is the most broad classification. So we don't use class at all. So order would be the biggest umbrella down to an individual species. But I did want to point out, you'll see these particular suffixes being used depending on whether you're talking about order or family or genus. So if you see something ending in viralis, that's always referring to an order. So Picornoviralis would be that particular order of viruses, whereas viridae, that suffix is always referring to a particular family. And then suffix virus is always referring to genus. And then we do have individual species, and this gets sort of complicated in the microbiology world, because within the species you've got different subtypes or strains or isolates. You know, there's lots of different terms that could be used, but basically different flavors of that species that may be genetically unique and even phenotypically a little bit unique as well. So anyway, when you're talking about a specific species, a lot of times you'll see nomenclature like this, right? So, you know, human rhinovirus 1 or versus human rhinovirus 1A or 1B would be getting down to different specific strains of that one species. All right, so I mentioned orders were the most broad classification, and here are the current documented orders of viruses. And I'm not going to go through them all, but I might throw 1 or 2 of these out on a test. But you can see in the notes here right? They're always mentioning, okay, what kind of genome do they have? Um, what kind of host do they infect? Is it plants or animals? Um, I'll mention Nidovirales here is the order that COVID-19 falls into, the coronaviruses. So they mention, this is a positive strand. We'll talk about what that means in coming chapters. Uh, Single-stranded RNA virus infects vertebrates and invertebrates. Um, this name Nidus uh, actually translates as nest, and that's because we have like nested genes within the genome. So you can have one piece of messenger RNA. It's actually getting translated to encode several proteins. Just to give you one example here. So I am going to end it there. It was a relatively brief chapter. Chapter 3 you are going to be responsible for on your own because it is mostly a basic review of basic biochemistry and molecular biology that you should have seen elsewhere as a biology major. So I'm just going to ask you to read chapter 3, and you're going to have to turn in the end of the chapter questions. And then when we meet again for lecture, we'll be talking about chapter 4.